All right, so we're going to go over questions in relation to the cell, cell division, cell parts, blah, blah, blah. Okay, so here are questions that we're going to go over. So here, first question is, what are the 3 points of the cell theory? And then the question is virus alive? So we're going to answer this question first. All right, some of the questions have already been answered just to save a little bit of time. Some of them haven't. So 3 points of the cell theory. First point is all living things are made of cells. Cells are the functional unit. So like, for example, if you think about motion, that actually comes down to certain specific cells, muscle cells, that actually contract to generate motion. If you're thinking about thought, the brain cells that are actually working together to carry out thought. So that means that when you think of a function, these are a group of cells that are working together to carry out that function. And the last one is all cells come from pre-existing cells. Of course, we have a bit of a breakdown. I mean, what that means is that you came from cells from your mom and dad, which came from cells from her mom and dad, which, keep going back all the way to the beginning. I guess the first question is all cells come from previous cells, but what about the first cell? That's an interesting question. What about the first cell? All right. That's obviously something that we're not going to address in grade 10 science. You'll learn probably about that maybe in grade 11 biology when you learn possibly about evolution. All right, so, and is a virus alive? Well, we can say the answer to that question would be no, because it is not made up of cells. We made that we distinguished that a little while ago. Okay. The next question has to do with cell parts. Now, I'm kind of proud of this video. I made a video a long time ago about a beta cell. So a beta cell is a specific cell in your pancreas that makes insulin. So the video goes over how the different parts of the cell work together to produce insulin, kind of like the video we saw with Inner Life of a Cell, the different parts working together. So if you're curious, if you want to watch that video clip, this is the link to it. I think I put the link also in your course in the bio folder, so you can take a look at it. I think it's my most watched video, I believe. So anyways, it goes over like, if you're going to make insulin, you need parts to work together. That's the idea, right? So parts got to work together, like the nucleus, the DNA, the ribosomes, the rough ER, the Golgi, the membrane, all these things got to work together to, make insulin and release it, right? Okay, anyways, so let's look at some parts of cells, right? What their job is. What are the functions of the following parts? So the cell wall, which is only found in, actually it's found in plants and bacteria. Fungi too. Oops. But we normally always just think about plant cells that have cell walls. And what do cell walls do? They provide protection. Whether it's plant cells, bacteria, fungi, a cell wall provides protection to the cell. Now, if you remember the cell membrane, the role of the cell membrane is to control, one of the roles is to control movement in the cell. But that was not the only function. We talked about the fact that the cell membrane is also involved in things like cell recognition, that the cell membrane is also involved in cells being able to anchor to other cells. So there's obviously more than just controlling movement in and out of the cell, but that was our focus in this section on the cell. Ribosomes make proteins. If you remember, there's two types. There's the ones made from the free ribosomes, and those are the proteins that stay in the cell. If you remember, the ribosomes could be bound as part of the rough ER, and those proteins, they exit the cell. So like, for example, for something like insulin, that would be protein that exits the cell. So it would be made from something like the rough ER, okay? Whereas like, for example, like an enzyme, let's say, you know how we talked about lysosomes and how they break down stuff. So let's say a protein, a digestive enzyme that's in the lysosome, that would obviously stay in the cell. So that would be made by like something like a free ribosome. Just give you an example of each one. Okay, so then the ER, we can divide it into two parts. The rough ER, we actually just talked about that. The rough ER is what makes proteins that leave the cell. So there you go. We actually already addressed that. Whereas a smooth ER, so it has no ribosomes. So it looks kind of smooth in appearance. They have other roles. So they don't make protein, but they make fats. For instance, phospholipids, so parts of the cell membrane. They make cholesterol. If you remember, cholesterol is a cell membrane stabilizer that keeps the cell membrane not a solid, not a liquid, but it's only found in animal cells. If the smooth ER is found in the liver, it's also involved in detoxification. In other words, getting rid of things that are poisonous. The next organelle is the mitochondria. So the mitochondria is the organelle that has actually two membranes. So it would have two bilayers, an outer one and an inner one. And you'll learn possibly why in grade 11 again. It's an interesting story behind the mitochondria. So what does it do? It uses oxygen and food, specifically sugars, to make energy, and waste. And the waste actually is in the form of CO2. So we think we talked about the mitochondria being like the battery of the cell. All right. The next organelle is the lysosome. So it's involved in, you can think of it as the digestive system of the cell. So like basic breakdown of things that are swallowed by a cell. So if a white blood cell, for example, swallows a bacterium, imagine this is a bacterium, then the lysosome would then take that bacterium and break it up into little pieces and basically digest it. But it's also involved in cell death. That's what apoptosis means. The way it works is it has enzymes, degrading enzymes, not because it makes fun of the things that it's, yeah, that's a bad joke. These enzymes are special proteins, and their job is to break up things. Your digestive system uses the same or similar types of proteins when you eat food. Thing about this is that these work well in an acidic environment. So that means if you looked at the lysosome, so the lysosome, the inside of the lysosome would have lots of protons, because if you remember, we talked about acids and bases. the pH scale. When it's acidic, you have a lot of protons. The more acidic, the more protons. So the way it gets an acidic environment is it actually has a proton pump that actually brings in the protons from the outside of the lysosome, from the cytoplasm into the lysosome to make it an acidic environment. We also use this as an example to discuss a disease. If you remember, Tay-Sachs disease is due to, I guess you could say your lysosome is not working because there's a specific enzyme that actually is malfunctioning. And what happens in Tay-Sachs is the lysosome can't break down fats. So the fats accumulate, and that's actually a big problem for a developing baby's brain. And the condition is fatal. And I think we talked about that. Okay. The next organelle is the Golgi apparatus. This is also a structure made of membranes. You can see that. So what does it do? This is what, for example, would receive from the ER, would modify it, and then it would ship it out of the cell. So if this cell was a beta cell, then the insulin would actually arrive in a vesicle. The insulin would be then enter the Golgi body, get modified, and then eventually would get shipped out of the cell. The chloroplast. The chloroplast is kind of like the mitochondria. It's also a double membrane structure. So you can see it's got an outer membrane and an inner membrane. It's got two bilayers. And its role is to take light and carbon dioxide and other ingredients too, but let's just keep it simple. Light, carbon dioxide, and convert that, well, we can say, let's add the other thing, which is water, and convert that into sugar and oxygen. So you can see, by the way, there's a symmetry between the chloroplast and the mitochondria. If you remember, the mitochondria takes oxygen and sugar and makes energy and waste, and the waste being carbon dioxide. And you can see that the chloroplast pretty much does the opposite of that. So there is a symmetry there between these two organisms. Okay, the next organelle is the peroxisome. This is an organelle that kind of sounds like the lysosome, but it's got a more specific job. Its specific job is to break down specifically fats and certain proteins. And when it does that, it produces a poison called hydrogen peroxide, which is toxic. You might know hydrogen peroxide. I think you might use it for, like, I think it's used to bleach hair, if I remember correctly. You can also use it for, like, sanitization. Yeah, I'm sure you've heard of hydrogen peroxide at some point. You've come across it. Anyways, this is a byproduct of when lipids are being broken down. It's toxic. So what happens then is the peroxisome, now you can probably guess why they call peroxisomes, because they produce hydrogen peroxide. They use something called catalase. Catalase is an enzyme that then breaks down the hydrogen peroxide into water and oxygen, which are obviously safe byproducts. Now, the reason why this is happening in the lysosomes is because you are producing something toxic. You don't want the hydrogen peroxide to leak out in the cell. You want to keep it in a safe environment, right? So that's why it's done in these things called peroxisomes. Okay. Our next is the vacuole. Remember, a vacuole is found in plant cells. A plant cell is usually like a rectangular cell. The vacuole is actually quite significantly large relative to the size of the cell. And its job is to store water primarily. The nucleus's job is to, remember, protect the DNA. That's its job. That's the most valuable information inside the cell. Okay, so that was question 2. Question 2 was some random different organelles that we could look at. Question number 3 is, what are the differences between prokaryotes and eukaryotes? So we could say this. We could say that eukaryotic cells have a nucleus. We talked about that. But we can actually add a bit more to that. We can also say that they have membrane-bound organelles. So things like the mitochondria, things like Golgi complex, things like lysosomes. If you look at the lysosome, you can see the membrane. These are all organelles that have membranes, right? So these organelles, we see them in eukaryotic cells, but in prokaryotic cells, we don't see these types of organelles at all. They're much simpler compared to the eukaryotic cell. So that's actually another huge difference between eukaryotic cells and prokaryotic cells. So one is they don't have a so one is eukaryotic cells have a nucleus, bacteria don't. Eukaryotic cells have membrane-bound organelles, eukaryotic cells don't. Sorry, prokaryotic cells don't, okay? Bacteria do not have membrane-bound organelles. Okay, so, all right. Number 4, describe the organization of the nucleus. So the nucleus, we said, its job is to protect the DNA. That's its job. So what do we see in the nucleus? Well, the nucleus contains a nuclear membrane that controls what goes in and out of the nucleus. It contains a nucleolus. Nucleolus is the structure that actually makes ribosomes. So if you look at a cell and you see a nucleus, the nucleolus is but it's often you'll see there's a dark spot in the nucleus. That's the nucleolus. That's where the ribosomes get made. And then you have the DNA. Now, the DNA is going to be organized into chromosomes or chromatin. So if the cell is not dividing, then you will see it as chromatin. Actually, you won't see it because the chromatin is very thin. So this is when the cell is not dividing. So it's not undergoing mitosis. But when it is going to undergo mitosis, the chromatin condenses into chromosomes. So that's when the cell is going to undergo division, mitosis or meiosis. All right, that's the difference between the two. All right, next question is the cytoskeleton. So the cytoskeleton, we actually talked a little bit about it. These are basically proteins that they link up or disassemble. They can assemble or disassemble. Includes, if you remember, the spindle fibers. So these are the things that attach to chromosomes and move them around. So they're involved in certain functions like movement. If you think about your muscle cells, when they contract, that's actually the part of the cytoskeleton that's actually contracting. If you remember from the video Inner Life of a Cell, it's also involved in intracellular transport. That means transport inside the cell. And when a cell is dividing, if you remember, the cell will start to pinch in the middle. That pinching in is cytokinesis. That's the result of the cytoskeleton forming like a noose around in the middle of the cell to pinch it off. Okay, number 6, form and function. So form and function is when the shape correlates to what it does. So the red blood cell is round. That's so it can easily move through your blood, which is good. We want the red blood cells to easily move. Remember, the platelets, they're not round because we don't want them to easily move through the blood. They want them to actually be involved in blood clotting. If you remember, the other example we talked about was like the hepatocytes or liver cells. Remember, liver cells have a very, very visible nucleolus because liver cells make lots of proteins. So it would make sense that they would have a very visible nucleolus because the nucleolus makes ribosomes, which make proteins. The idea behind it is, if you know what some sort of business does, you can kind of predict what it would look like. Like if I know my business is involved in storing, like warehouse storage, then obviously my business needs a building that has lots of space for storage, right? However, if my business is involved in manufacturing, then I know when I walk into my building, I'm not going to see all this empty storage. I'm going to see lots of machinery in my business. So going back to the red blood cell, if I know that my red blood cell is involved in transporting oxygen, then I know that it also is going to have no mitochondria, because if you remember, the mitochondria uses oxygen. Therefore, it would make no sense for it to have mitochondria. Okay, that's number 6. In number 7, it's about cell death. This is another word for cell death, apoptosis. So what occurs when there's too much apoptosis? That actually leads to degeneration, where your body starts to break down because it's not healing itself. That's not good. But if there's too little apoptosis, then that could be or lead to cancer, because it has to be a balance, right? We want the cells to divide when there's, the right conditions for division. When there's death, we want it to divide when, for instance, an organism is growing. When there's damage, then we want cell division, right? But we want to balance it out, because if you don't balance it out, if there's too much cell division, then that could be a problem too. That could lead to something like cancer. Okay. Speaking of cell division, our next topic is on cell division. There are some animations in here, virtual cell, you can take a look at if you want to, review mitosis and meiosis. What is the purpose of these two things? So mitosis is to produce identical cells. And this is usually for growth and repair. Whereas meiosis, remember, those produce your sex cells. So they are not identical, because the whole point of sexual reproduction is to produce offspring that are different. So they have a totally different purpose, okay? If you remember in mitosis, the stages are prophase, metaphase, anaphase, and telophase. Prophase is when you first see chromosomes. Okay. Metaphase is when the chromosomes line up, so they're in the middle. Anaphase is when they pull apart. And telophase is when you can see the nucleus is reforming. And that would be the end of cell division, right? That's when then you'll start entering into the interphase, in preparation maybe for another round later on. Cytokinesis. So after telophase, when the cells actually you start to see the nucleus reforming, the so the chromosomes now are on either end, right? And then you can start seeing the nucleus is starting to reform around it. What happens is the cell, if it's an animal cell, will start to pinch it in the middle. Okay, that's called cytokinesis, right? And the reason why that happens is because we want to eventually split these cells into two cells, right? That's why we do that. Now, remember, it doesn't happen in plant cells. In plant cells, they form a cell plate because the cell membrane can't pinch. Sorry, the cell wall can't pinch. Remember, plant cells have a cell wall. So instead, what they do is they form what's called a cell plate. Okay, so that's a slight little difference there. All right. The next question, my numbering is not great. I have 3 twice. My next question is in relation to meiosis. So what happens in meiosis, if you remember, and the same thing actually happens in mitosis, is before anything, the DNA is copied. So in both mitosis and meiosis, the DNA is copied, right? So let's say I have a chromosome, okay, and then I copy it. Now I have an identical copy. Okay. So what do we call these identical copies? These are actually called sisters or sister chromatids. Okay. Now, here's the thing. Because you have 2 pairs, you have a pair from your mother and a pair from your father. If, let's say, okay, so let's say we have, you have 23 chromosomes, all right? And they're numbered number 1 to number 23. So what makes them, they're ranked by their size. So let's say, for example, chromosome number 1. Chromosome number 1, you would have 2 copies of chromosome number 1. You would have 2 copies of number 2, and so on and so forth. Where you might have differences, number 23, these are the sex chromosomes. So females have 2 copies of something called X, whereas a male only has 1 X and 1 Y. So let's look at chromosome number 1 to 22. These are called the autosomal chromosomes. You don't need to know this term, okay? Males and females are exactly the same. So if I look at chromosome number 1, I have 2 copies of chromosome number 1. I have a female version and a male version. So like we said, so here's, imagine that's your female version. And this is your male version, okay? So here's your chromosome. And what happens is we know that the chromosome gets copied. So now they become sisters. So there you go. There are, now I copied my chromosomes. Okay, now, if they are the same type of chromosome, so if these are both chromosome number 1, that's chromosome number 1, and that's chromosome number 1, then we say the chromosomes are homologous. Okay, so homologous chromosomes are the same type of chromosome. It's just one is from your maternal side and the other one is from your paternal side. Okay, so that's what homologous chromosome. So the same type of chromosome, just one is from your mother, one's from your father. Whereas the sisters are identical because they're copies of each other. Okay. What is the function of meiosis? Well, we actually repeated that. We already discussed that. So that answer was over here. The idea was in meiosis, actually it's right over here, we want to produce sex cells. All right. So there's a bit of a repeat there. What is a tetrad and what is crossing over? We can actually address these 2 questions together. So when you have these homologous chromosomes, you'll notice that there's 4 of them, right? Because we started with 1 of each, 1 from your mom, 1 from your father. We made a copy of each one. Those are your sisters. So now we have 4 chromosomes. So what happens then is, let's say that this is your mother's set that was copied. So there are your 2 sisters. All right. And then the father's set was copied. Those are the 2 sisters. Now remember, if it's the same chromosome, so if these are both chromosome number 1, they're homologous. But what happens in meiosis is that they come together and they form a structure called a tetrad. Think about the term tetrad, it's 4, right? So they come together. Now, so a tetrad is the homologous pairs come together. All right. Now, why do they do that? If you remember, they do that so they can exchange DNA. So if I have, for instance, let's imagine I have a crossing over event right here. Let's imagine these sections cross over. And what that does is the blue and the red, that section gets swapped. So what that produces then is something like this. Okay, so you can see that after the crossing over, we've actually, swapped some DNA. Now, the reason why, so this occurs between the homologous pairs. There's no point in you doing this between the sisters because the sisters are identical. So what does this do? This produces a new combination in your chromosome. The idea here is that in sexual reproduction, you want to generate a lots of variety, right? It's actually one of the next questions coming up. So sexual reproduction, the key is to generate variety, produce offspring that are very, very different. Okay, so this is actually a mechanism that generates variety in sexual reproduction. Okay, compare meiosis to mitosis. What are some of the key differences? We actually already addressed kind of that, but just to recap, so key differences. So in mitosis, the cells are identical. So in meiosis, they're not identical. So why are they not identical? Well, one, because we have crossing over. Okay. The other is because if you remember in meiosis, you actually have 2 rounds of division. So meiosis, you have the cell will split into 2 cells, but then they'll split again into 4. And when they do that, so you have a second round of division. So that's another difference. There's 2 rounds of division. And the daughter cells will have half the DNA at the end. And if you remember, the reason why is because we want to produce sex cells. So the sex cells have to have half the DNA, so that when the sperm cell, which is half the DNA, meets up with an egg cell with half the DNA, then you get a full complement of DNA. So then we can actually answer this question, because if we think about the variation that sexual reproduction produces, the variation is due to things like crossing over, because crossing over mixes up the chromosomes. It's due to generating sex cells with half the DNA. But if you remember, the idea was because there are so many possible combinations, I think it was like about 8 million possible ways of doing this. Let me maybe show you this, what I mean by that. So the DNA is, you're only getting half, the sex cell only gets half the DNA, right? So if I have, for instance, imagine I have something like this. I'm going to call this A1, A2, and I say B1, B2, okay? Imagine something like this, okay? All right, imagine that set A is mom's set and set B is dad's set, okay? So chromosome number 1, I have two versions. I have one from mom, one from dad, A versus B, right? And I have the same thing for chromosome number 2. I have A from, let's say A was from dad and B was from mom, whatever, okay? Now, the daughter cells can only get half the DNA, right? So at the end of meiosis, you're either gonna get something like this. You're only gonna get one of the ones. You're either going to get A1 or B1. You're not going to get both. You're just going to get one. So let's say you get A1 with, let's say, B2. Okay, so you'll notice that for chromosome number 1, I only have one of the two versions. And for chromosome number 2, I only have one of the two versions. So I have half the DNA. But mathematically speaking, how many possibilities can I generate? Well, it turns out that the mathematical formula for determining this is 2 to the power of the number of chromosomes, which is in this case 2 to the power of 2, right? Because there's two chromosomes, chromosome 1, chromosome 2. So the possibilities would be 4. 2 to the power of 2 is 4. I can get A1, B2, I can get A1, B1, I can get B1, B2, and B2, B2. Those are 4 possibilities, right? With 23 chromosomes, it's 2 to the power of 23, which would produce 8 million combinations of sex cells. Then when you have fertilization, you know, that means if a female can produce 8 million types of sex cells and a male can do the same thing, then the random combination of that would generate 64 trillion possible zygotes. And that again is just without crossing over, right? So you can see how the variation is generated through this process. And that also, by the way, doesn't even include one more thing, which don't really do too much in grade 10, and that are mutations that arise. These are just common mistakes. Well, they're not that common, but they're mistakes that happen when, you know, cells divide or when DNA gets copied. Okay, so we covered the cell, cell parts, cell division, and then the last section was cell transport. Okay. All right, so this last section is on moving things in and out of the cell. This is the last thing that we did. So here's the first question. What is diffusion? So diffusion is, it's a natural movement of, so no energy is required. Let's write that down, no energy required. Okay, we say it's a natural process. Things are moving from a high to a low concentration. Okay. What are substances that can diffuse into and out of cells easily? So things like gases and water, although the diffusion of water actually is called osmosis, and lipids. Now, why lipids? Because if you remember, the cell membrane is made of fat, and in chemistry, like dissolves like. So things made from fat can dissolve through the cell membrane. So for instance, as an example, let's say you make, let's say this is the cell, and the cell membrane is made of phospholipids, so it's primarily fat. So it's pretty much a lipid membrane. Estrogen, you might know of what estrogen, you might have heard of estrogen. So estrogen is a hormone which is made of fat. Okay, it's a lipid-based hormone. Now, whereas insulin is not. Insulin is actually a protein-based hormone. So if you remember, insulin actually has an insulin receptor, and that's on the outside. And that's because insulin just, can't go through the cell membrane. So it needs a receptor on the outside. But estrogen is made of fat. So estrogen can actually go right through the cell membrane. Okay. So gases, water, and things made of fat will go right through the cell membrane. Right. So osmosis is the diffusion of water. Okay. If you remember, the water will diffuse, moves to the hypertonic side. So what does that mean? Right? Because that's actually the next question. So hypertonic means it has a lot of solutes. Hypotonic means it has low solutes. And isotonic means the solute is the same on both sides. So solute is equal. So for example, if I have a beaker, maybe use this example, and let's say we have a cell in water, and the water or solution, and the solution has a 5% salt concentration, and the cell has a 1% salt concentration, then we could say that the inside of the cell, we could say that the ICF, the inside of the cell, relative to the outside would be hypotonic. Whereas the extracellular fluid outside the cell would be hypertonic. Now, why is this important? What does it have to do with the health of the cells? Because if too much water leaves the cell will shrivel up, and it will be very hard for things to move through the cell. Reverse is true. If the cell fills up with water, then the cell could burst, and that wouldn't be good either. So the amount of water in the cell is very, very important into maintaining its health. All right, we're almost done. The next question is active and passive transport. So if you remember, active means energy is required. Passive is no energy. So diffusion would be an example of passive. Whereas as soon as you hear the word pump, that means we know that it's required, energy is required, okay? In secondary active transport, that means you have some sort of a pump connected to facilitated diffusion. So the example that we saw in the video clip was, imagine that this is the membrane. You have maybe a pump, like possibly the sodium potassium pump, the NAK pump. And then you have a co-transporter that allows for sodium and maybe glucose to diffuse through the transporter. So what happens is it's called secondary active transport because the first part is active. So this part's active, but the second part is passive, right? So that would be an example of secondary active transport. Next question is the cell membrane is semi-permeable. What does this mean? You might actually hear the word selectively permeable in certain books. So what does this mean? That means some things can go through. So the key is some, which obviously means some things can't. Okay, that's what that means. If it's semi-permeable, some things can go through, some things can't. All right. If you can't go through, then the only way they're going to get in are through doorways, special proteins, like, for example, channels. A channel is a special protein. So this is an example of facilitated diffusion. Okay. So if they can't go through the cell membrane, like normally on their own, then they'll have to go through a doorway. If it's a channel, channels are things that allow ions to go through, things that are charged, okay? And these would be examples of passive transport. They don't require energy directly. So this would actually also be an example of, but this is not a channel, because a channel is not, because glucose isn't charged. We would call this simply facilitated diffusion. You can call it maybe a carrier protein. But this would be an example of a co-transporter that does not require energy directly, but it's also requires sodium, which needs to be pumped out. So that one would require the sodium potassium pump to make it work. So that's secondary active transport. But the actual channels and carrier proteins, things involved in facilitated diffusion, they're all directly passive. They don't require any energy. Are pumps, well, we actually just answered this question. Are pumps active or passive? We just answered that. They are active. Okay, they require energy. And we actually already just answered the last question. What is secondary active transport? And the example that we just did is sodium glucose. So we actually already just addressed this question. What is secondary active transport? That means that your transporter that works by facilitated diffusion requires a pump to make it work. So the transporter itself, the sodium glucose transporter, is passive, right? It is passive, but it won't work unless sodium goes through it. Sodium needs to be pumped out, which requires a pump. So therefore, it's considered secondary active transport. Wow. Okay, that covers everything from the beginning, which was the cell parts, cell division, and lastly and leastly, cell transport. All right, let me stop recording and take any questions if you have any.